All right. Again, thanks everybody for being here. I hope it was a wonderful and restful weekend for you all. Certainly, we have a lot to get covered this unit and today, and we're going to hope to finish up this unit this week. Things to look forward to. Wednesday is a half day, so the schedule will be a little bit different on Wednesday. I will talk more about that tomorrow. Uh, Teachers will be back in the building, the school building, on Thursday, so you'll see me with a slightly different background, and there might be some trial and error with that, and I hope you all can be patient and gracious with me as I try to figure out, what teaching virtually, but also students in person looks like. So that'll be an interesting experiment on Thursday and Friday. Then on Monday, some of you hopefully will be joining me in the school building, and it'll be nice for me to be able to see your faces, if only the top half of your faces, and it will also be nice for me to be able to interact with you all and draw things on a whiteboard and just get some more interpersonal connections, and hopefully being able to talk with you and seeing your eyes and seeing what you're understanding and not understanding will go a long way in terms of our progress in the course. But I know some of you have opted to continue learning virtually throughout the school year, regardless of the fact that we're going back in person. And I understand that 100%. So we're going to have to just continue to be creative and resilient as we think about ways to make sure that you don't start to fall off because something is that opportunity won't be there for you all to be there with me in person and for me to be able to really engage with you all. But things are going to get different over the next week or so, but we're going to work together. This will be a collaborative process. I want as much feedback as possible. So if you all are frustrated by the way something is going, or if we find that my teaching while doing so both virtually and in person is not effective, I want you to let me know. And certainly I'm willing and ready to make changes when and where those changes will be necessary. So just keep that in mind. It's going to be an interesting transition. It may at times be frustrating. It may at times feel like a step back, but we will ultimately get to a point where I think it will feel a little bit more natural. So for those of you who are in that first rotation, I look forward to meeting you a week from today. And everyone else, those of you who might be in a later rotation, and those of you who are not returning, who will be virtual, certainly I still look forward to working with you all as well. Keep in mind, though, those of you who have signed up to come back in person, which is the majority of you, if you do not, then unfortunately I will have to mark you absent. That's not my decision. I think it's a bad policy, but at the end of the day, that's what we're being told we have to do. So those of you who are supposed to be in school, in order for you to be, even if you choose not to attend, but you are still logged into the live session, I will still have to mark you absent. At least that's the direction that we've been given most recently. That might change as time goes on, but and I hope it does. But for now, we're just being told that if you're supposed to be in person and you're not in person, then you're absent. I think it really is a liability issue because if you're supposed to be in person and you're not, then we really can't account for your well-being. And so it wouldn't be right for me to mark you present, though I cannot actually say whether or not you are here. I agree, Jason. It's a bad policy, but it's not one that I made. It's not one that our school has made. It's something that the district has decided. So, like I said, I think it's a liability thing, but we work with what we're given. That being said, today is the second day of our fourth unit. We're going to discuss a lot today, so I need you guys on your A game in terms of asking questions, answering questions, and in terms of taking notes. We've got a lot to cover. I know you all are ready to do it. This is the wrong objectives. Give me 1 second. All right. Sorry about that. Needed to fix that really quickly. So our objectives today, we've got two things. And of course, this is because there are two topics. The carbon cycle topic is pretty short, which is why I've decided to combine it with the cell membrane lesson. The first objective is to understand how carbon flows both within and between systems. So within a system, for example, within your body, how does carbon flow? But then also between systems. So how does carbon go from our bodies to then the atmosphere, and then to trees, and then to the soil, and then to another animal, and then back to our bodies? So we need to discuss these things and why this flow is important. The second objective is to understand the properties of the cell membrane. The cell membrane is really, really cool. We obviously talked about the cell membrane as a part of our second unit on cells, but the cell membrane has so many special properties that we also have to really define and discuss in detail today. We do have an essential question, two of them. One of them states that, or asks, why is it important that carbon can be transferred? The second one asks, why does the cell membrane, it says how does the cell membrane function? Excuse me. So again, strong note takers, start here on this first slide. By the end of today, this gives you a goal, this gives you a topic, this gives you a date. All of these are things that allow you to return to your notes and use them in your studying process. Learning is not just simply sitting here and listening. Learning is engaging with the material. Learning is asking questions of your notes. Learning is drawing diagrams. Learning is adding colors and highlighting and underlining and circling. So learning is an active process. It's not something that's passive. It's not something that you can just sit and listen. That might work with a podcast or a video, but typically it's best when you are actually experimenting or experiencing something yourself. All right, so before we jump into it, you guys know I like to start Monday with some lessons outside of the classroom, lessons outside of science, and also just to give you some motivation to begin your week on a strong foot. So in honor of yesterday being Valentine's Day, and in honor of the fact that we're still in Black History Month, we're going to talk about a story in which love conquered racism. And this is the story of Loving v. Virginia. So in many southern states, of course, North Carolina is considered a southern state, has been a southern state. There were laws that prohibited many things, that prohibited a lot of freedoms. So Black people were not allowed to do certain things that white people could. And Black people were also monitored in ways that white people weren't. For example, there were vagrancy laws that prevented Black people from not having a job. They were required to not only have a job, but also have proof that they were employed at all times. So little things like that, that because of the weight of all of those effects, the cumulative weight of those effects, really limited the freedom of Black people. One of the laws that did such was a law in the state of Virginia called the Racial Integrity Act of 1924. And this law prohibited interracial marriage. It prevented people of color from marrying what was referred to, or who were referred to, as pure white people. It established that only people who had absolutely no trace of Blackness in their ancestry could be considered white. And it also required that on people's birth certificates, their race be recorded. So this meant that when you would and applied for a marriage license and had to produce your birth certificate, the state would be able to say, okay, these are two people who were marked as colored at the time of birth, so they are allowed to be married. These were two people who were marked as white at their time of birth, so yes, they are legally allowed to be married. Or this person is marked colored, this person is marked white, it's illegal for them to be married. So this simple act, some people might consider a basic freedom of marriage, was being prohibited. Okay, this couple called the Lovings. There was a woman named Mildred Loving. She was a woman of mixed ancestry. She had Native American heritage or ancestry, as well as African American ancestry. You can see her image here. And a man named Richard Loving, he was white. They grew up in the same community. They were essentially neighbors for their entire lives. They attended the same schools. And in high school, they fell in love. Mildred ended up becoming pregnant in 1958. And if you know anything about that time in American society, of course, when a woman got pregnant, it was almost expected that the man would then marry her. But of course, they were in love, so this didn't present a problem to them. They ended up leaving Virginia to go to Washington, D.C., where interracial marriage was legal. And in Washington, D.C., they were able to obtain a marriage license. They returned to Virginia, and later they were arrested. Not only had they broken the law because they had become married, but also because they were traveling together. So at the time, in many southern states, white and black people were not allowed to cross state lines with one another. That was also considered illegal. So the police raided their house in the middle of the night, hoping to catch them, in the act of being together and having more evidence against them. They found the loving couple sleeping in their bed, and even though Mildred was able to reproduce a marriage license, she was able to point it out to the police. They considered it void and null because it was illegal for them to be married in Virginia. The Lovings were arrested. They had the possibility of being sentenced to up to 5 years in prison. They were convicted and sentenced to only 1 year in prison, but that sentence was suspended if they agreed to leave the state of Virginia for at least 25 years, which they did agree to. They ended up moving to Washington, D.C., where they lived basically, in poverty and lived away from their families, lived away from their livelihoods, lived away from the places where they had been educated and where they had begun their careers. So, of course, this contributed to considerable homesickness. So at one point, a few years later, I think 5 years later, Mildred decided she was going to return to Virginia only for a visit to see her family. And upon doing so, was arrested again. And so the second time that they were arrested, this couple was arrested, they decided to appeal this conviction. They decided that they weren't just going to allow themselves to be punished again. They decided to appeal this conviction to the Virginia Supreme Court. Those of you who are interested in law and you think you might want to become a lawyer, these court cases, what happens is that when a person is convicted of something, they often will have the option to appeal that conviction. And when you appeal the conviction, what you're doing is saying you want another set of legal eyes to take a look at this case, to make sure that everything was done properly, to make sure that nothing that happened was unconstitutional. And you want maybe another group of lawyers, like a Supreme Court, a state Supreme Court, to take a look at this conviction. That's what they did. The Virginia Supreme Court upheld the conviction, meaning that they said, nope, we checked and everything was done properly. Unfortunately, your conviction is going to stand. But the Lovings lawyers were not satisfied with that. They felt that this was unconstitutional, that preventing white and black people from being married was something that, limited the constitutional rights of people of color. And so what they did was they appealed it again, but this time they appealed it to a higher court. Oftentimes the higher court might be a district court, which is a court that basically oversees several different states. But instead of doing that, they appealed to the highest court, the Supreme Court of the United States. This is the court that makes final decisions. If the Supreme Court decides one way or another, that's it. There is no more appealing. So they basically took this one last chance, and this is what happened.
On July 11th, 1958, at 2 a.m., a county sheriff entered the home of Virginia couple Richard and Mildred Loving, forced them out of their bed, and placed them under arrest. Their crime? Interracial marriage. This is the story of Loving versus Virginia and the landmark civil rights decision that would set an important precedent for marriage rights in America. Richard, who was white, and Mildred, who was black and Native American, spent the early morning hours of July 11th in jail. They were tried and found guilty of violating Virginia's Racial Integrity Act of 1924, a series of laws that made race mixing illegal. The Lovings were then given an ultimatum: spend 1 year in prison or leave the state of Virginia for 25 years. Frightened and unaware of their legal options, they moved to Washington, D.C., where their marriage was legal. They raised their 3 children in the nation's capital, financially strapped and isolated from friends and family. 5 years later, they returned to Virginia to visit Mildred's relatives, but were arrested for a second time for traveling together. Inspired by the burgeoning civil rights movement, Mildred decided to fight back. So in 1963, she wrote a letter to U.S. Attorney General Robert F. Kennedy asking for his advice. Kennedy referred the Lovings to the American Civil Liberties Union, who assigned their case to 2 young lawyers named Bernard Cohen and Philip Hirschkopf. Cohen and Hirschkopf took their appeal all the way to the Supreme Court, arguing that Virginia's anti-miscegenation laws violated the Equal Protection Clause of the 14th Amendment, which guaranteed African Americans citizenship and all its privileges. The broad and sweeping language of the amendment, they argued, made no mention of interracial marriage, and thus guaranteed equal protection of a human being's right to marry. Attorneys from the state of Virginia argued that the state's rights should be prioritized over federal laws. But the argument didn't hold up. On June 12, 1967, the court unanimously struck down the Virginia marriage ban and overturned the Lovings' conviction. The court ruling reverberated across America, invalidating anti-miscegenation laws not only in Virginia, but in 16 other states that held them as well. After winning their case, the Lovings were finally free to do what they wanted to do all along. They lived quietly in Virginia as husband and wife until Richard's death in 1975. Loving versus Virginia would inspire gay marriage advocates during their own Supreme Court victories decades later. For example, 2003's Lawrence on July.
Okay. And I will close off this segment of class with this quote from Mildred Loving. She said, "I am proud that Richards and my name is on a court case that can help reinforce the love, the commitment, fairness, and the family that so many people, Black or white, young or old, gay or straight, seek in life. That's what loving and loving are all about." Okay, so I like this, and I use it as motivation for this week because not only was yesterday Valentine's Day, and it's nice to feel surrounded by love and to be reminded of the power of love, that love was able to propel this couple to fight for, what they believed in. And in fact, it ended up resulting in rights being granted to hundreds of thousands, if not millions of Americans who wanted to be able to marry whoever they wanted. But I also bring this up because it's important to acknowledge that the things that we take for granted in the year 2021 were things that people had to fight for. They were things that required groups of lawyers and required years of legal review, that also required people to be punished, to be imprisoned in the case of this couple, and in the case of several other couples. So these things that we take for granted, the opportunity to love who we want, the opportunity to marry who we want, the opportunity to travel with whom we want, to live with whom we want, the opportunities to be educated by whom we want. These were things that certainly we should not take for granted, because there are people who are still alive today. This was not very long ago. My grandparents are just as old as Mildred Loving, who fought for these things to be a reality for us. So yes, we can go through life and choose to ignore the history that got us to this point, but we should be motivated and inspired by the fact that even though we might still have a long way to go, we have things that we want to achieve as a society, but also as individuals, that will require hard work and sacrifice. We can at least be compelled by the fact that people have come before us and been able to do similar things, to sacrifice in similar ways, to give of themselves for the greater good. So keep that in mind, guys, and go into this week being inspired by this story. And those of you who are interested in law, we can certainly discuss more. As the video began to discuss, this case had huge implications on other cases as recently as 2015. So keep that in mind. All right. So not yesterday, but Friday, we talked about aerobic respiration. Does anybody remember what the word aerobic means? Thanks for joining us, Cindy. Okay, that word aerobic, hopefully you guys have your notes out and you're ready to go and you can refer back to your notes. But that word aerobic means involving oxygen. It means oxygen is involved. So we've got this equation at the bottom. This is the equation that really summarizes aerobic respiration. We can see that oxygen, O2, is being used. It is one of the reactants in this equation. So this is aerobic respiration. So what do we think anaerobic means? What do we think this word anaerobic means? If aerobic means involving oxygen, then what does anaerobic mean? Not necessarily exhaling. Involving does not mean to inhale. Involving just means that oxygen is a part of this equation. So aerobic means that we are using oxygen. What does anaerobic mean in that case, Jason, or anyone else? Kind of already on the screen. Thank you, Abraham. Yeah. Anaerobic just means the absence of oxygen. It's not being used. So I told you on Friday that there were different types of respiration. We really only introduced on Friday aerobic respiration, which uses oxygen. Today we're talking about a couple other types which do not use oxygen. They are considered anaerobic. When we do not use oxygen, we're not creating nearly as many ATP molecules. Instead of creating 36 ATP molecules when we do use oxygen, this time we're only creating 2 ATP molecules when we're not using oxygen. So not using oxygen is very inefficient. It doesn't work nearly as well as using oxygen. The 2 types of anaerobic respiration that we will discuss are alcoholic fermentation and lactic acid fermentation. So just keep in mind, guys, those of you who are athletes or who like to work out, when you are engaged in some type of exercise and you're working really hard, your heart rate is going up, your respiration is going up, it's good to take in as much oxygen as possible because that will help you to create more energy. If you're just relying on your body to do these anaerobic processes that do not use oxygen, they're not going to be nearly as efficient. All right. So lactic acid fermentation. Lactic acid fermentation only occurs in organisms that have muscles. But this happens when the muscles are not getting enough oxygen. And so as a byproduct, they create what's called lactic acid, and this causes cramps. So I know some of you probably have experienced cramps in various places in your bodies, but they are not a pleasant experience. Really, the best way to get rid of them is just to breathe. Give your body as much oxygen as possible. Stretch out your muscles. You might want to kind of, roll them out, your muscles, so they can get a little bit more oxygen. Deliver as much blood to the area as possible by stretching. But lactic acid fermentation occurs without oxygen. So the best way to cure these cramps is to give oxygen, to give your body oxygen. The second type of anaerobic respiration that we will discuss is alcoholic fermentation. And this occurs in yeast and bacteria. This process converts sugar into ethanol, which is alcohol, as well as carbon dioxide and ATP. So ethanol, of course, is what we use to make various, adult beverages between beer and wine and liquors. Ethanol is actually the active ingredient in those drinks. And then carbon dioxide is, of course, a gas. It's a gas that we, breathe in from the atmosphere, but it's also the gas that is used to produce bread. It's what causes bread to rise up. So again, both of these two processes, lactic acid fermentation and alcoholic fermentation, take place without oxygen. Okay. So here are the equations again, guys. Photosynthesis requires sunlight, water, and carbon dioxide. Those are the reactants of photosynthesis. As products, it produces oxygen and glucose. This is really what we want to focus on with photosynthesis. The goal is to use sunlight to make carbohydrates, to make glucose. Aerobic respiration is the opposite of that. Keep in mind, aerobic means involving oxygen. So in the reactants over here, we're just going to take these two things and put them down here, oxygen and glucose. And the goal of respiration is, of course, to make ATP, to make chemical energy. And so we're going to use oxygen and sugar as reactants to produce carbon dioxide gas, ATP, which is energy, and water. So please make sure that you have these equations down. We did show them on Friday, so I don't want to leave them up for too long, but I will leave them up for a brief period of time. Okay. We're not going to do this right now because we've got a lot to cover. We did that already. So then what does this all have to do with the carbon cycle? So the carbon cycle just tracks how carbon moves, like I said, both within and between systems. So in our bodies, we take in carbon dioxide. We breathe it in from the atmosphere, but it's actually poisonous to our bodies. So we need to be able to get rid of it. So when we breathe in air, we only want to keep the oxygen. Our blood needs the oxygen to be able to deliver it to our various cells. What we don't want is the carbon dioxide. So we breathe that back out. We put that back into the atmosphere. And all animals do this. Carbon dioxide is produced via respiration. It's one of those products of respiration. But anytime your cell is creating energy, anytime it's doing respiration, it's also creating carbon dioxide, and we need to get rid of it. It's a waste product. We breathe it out. It returns to the atmosphere. And once it does that, organisms that do photosynthesis will then use it because they need carbon dioxide to make glucose. They need carbon dioxide to do photosynthesis. So we've got this cool cycle where the things that we don't need, the things that animals do not need, plants do need. We don't need carbon dioxide. It's bad for us. Plants do need it in order to do photosynthesis. They take the carbon that's a part of carbon dioxide and then they transfer it. They make it into glucose. Additionally, we can think about other sources of carbon. Some of them are not so good. For example, when we use energy to power our connection that is allowing me to talk to you right now, but also to power everything else in our homes and in our schools and in our gyms and churches and our cars. What we're doing is we're burning some source of carbon. Oftentimes that source of carbon is natural gas, or it could also be oil. It could also be coal in some cases, but it's essentially some source of carbon. When we burn it, we're putting a lot of other carbon sources into the atmosphere. Some of them are not good. And this is what is leading to global warming and has been leading to global warming for the last 200 years or so. Another source of carbon that's, a part of this cycle, when animals die and when plants die. So when leaves get shed in the fall and they fall down to the ground, eventually they will decompose. Maybe over the course of the winter, maybe it might take a year or two, but eventually they will decompose. And all of the nutrients, all of the carbon that was once a part of that leaf can then become a part of the soil and lead to something else growing out of it. Same thing happens with animals. When animals die, parts of their bodies might be scavenged by other animals or by insects or by fungi, but parts of them will just decompose and become a part of the soil. And in doing so, they make the soil healthier so that other plants can then come and grow out of it. So decomposing matter is also going to give off carbon. And then even the waste from animals. So cow waste is a huge source of methane, which is also not good for the environment. So, there are vast portions of not only our country, but Argentina and South America and many West African countries and many Eastern European countries where vast portions of the landscape are dedicated solely to cows because we need their beef, we need their milk. And so because they take up so much land, they're also producing, they also produce a lot of waste. And their waste, this literal cow poop, produces methane, which is bad for the environment. So if you are a meat eater, if you like hamburgers, or if you like, what else comes from cows? If you like steaks, then you also, or if you like milk, that's the source of dairy. If you like ice cream, these are all products that come from cows. Unfortunately, the way that our current system is set up, using all of this land, giving all of that food to cows, allowing all of that waste to then produce methane, which is basically carbon, is not good for the environment. So we've got to find a healthier way of doing it. We're simply using too much land for cows. We have too many cows. So we've got to figure that out. That's a problem. Okay. So why might a plant at the bottom of the rainforest benefit from the adaptation of very large leaves? So think about being in the rainforest. You've got these super tall trees, but then you've also got smaller trees and maybe even some bushes and plants that are maybe only 2 feet tall. They sit on the ground of the rainforest. Why do they benefit from having larger leaves? What do you all think? Is it A, to get more sunlight, B, to access more water, C, to produce more carbon dioxide, or D, to stop them from being eaten? Why do they benefit from having really large leaves? Okay, we've got Jason saying C, as well as Brianna, to produce more carbon dioxide. Anybody else want to throw in an answer here? All right, so the answer is really not about producing carbon dioxide, because plants of all shapes and sizes can produce carbon dioxide. What we want to focus on is that having larger leaves allows you to soak in more sun. And when you're at the bottom of the rainforest, you've got this, what's called a canopy on top of you. All those really, really super tall and old trees have their own leaves that are kind of blocking the sun. They're soaking up most of the sun. So by the time you get to the floor of the rainforest, it's a pretty dark place. It's not like it looks like nighttime or anything like that, but it's very shaded. So in order for these plants at the bottom, sitting on the ground of the rainforest, to get any sunlight at all, they've got to cover a lot of surface area. So they want the biggest possible leaves to give them the greatest chance at getting some sunlight. That was just a thought question, really. So we can see that carbon moves from one process to the other. This process on top is photosynthesis. I know that because we're producing glucose, C6H12O6. So the carbon that's a part of glucose was at one point carbon that came from the atmosphere, carbon dioxide. And then when we go the other way and we're using carbon to make ATP, the carbon that was once a part of glucose is going to become a part of the carbon dioxide that then gets returned to the atmosphere. And it also becomes a part of ATP as well. ATP has carbon inside in it. So carbon is, like I say, it's staying in the system. It's just a cycle. There are a couple of videos that are a part of the asynchronous assignment for today that you all can watch to help you better understand this cycle, but just a little bit more information about it. Of course, photosynthesis requires energy from the sun. It also requires carbon dioxide and water. Photosynthesis is going to produce two things, oxygen and glucose. Those two things are then needed by all living organisms, not just plants, not just animals, not just eukaryotic organisms. All living organisms need to be able to do respiration. So they need oxygen, they need glucose. Respiration uses these two things to produce cellular energy, ATP. But it also, as a byproduct, produces carbon dioxide and water. And those two things can then again be used by photosynthesis. So we really have this cycle. The things that are produced by photosynthesis get used by respiration. And the things that are produced by respiration end up being used for photosynthesis. So there's this cycle going on here. All right, jumping into the next topic. Again, you're going to see some highlighted words here. Those highlighted words are the words that are super important for you to write down. This topic of homeostasis is a topic that we will stick with for our fourth unit. We'll talk about homeostasis for the rest of the week. Homeostasis is defined as maintaining a constant and stable internal balance. So internal, inside of your body, or inside of a plant, or inside of a single cell. Each cell within your body needs to have some balance. It doesn't want to stray too far away from its balance. We can think about temperature in human beings. Our ideal temperature is about 37 degrees Celsius, which is about 98, 99 degrees Fahrenheit. So inside of your body is pretty hot. Think about it, 99 degrees. If it were 99 degrees where you're sitting right now, you'd probably be pretty uncomfortable and sweating. But inside of your body, that's the ideal temperature. So if we get a little bit hotter than that, we say that you become febrile. You have a fever. That's not good for your body. It can actually cause your internal organs to start shutting down. Same thing, if it gets too cold, you can enter a state of hypothermia, where your body is going to start to direct this energy to protecting your internal organs. And your fingers and your toes will start to become frigid. They start to lose their blood circulation, and the tissue can rapidly die off. So we need to kind of stick in that middle range, between 97 and 100 degrees. That's where you want to be. Anything above or below that, you're starting to get into some danger zones. Same thing with your water percentage. About 70% of the human body is water. When you take into account your organs and your blood and your muscles and your skin, most of it is water, 70% of it. When you become dehydrated, you've got less than 70%. Again, your internal organs can start to shut down. Water is a necessary reactant in many chemical reactions. We need water. Our cells need water. And if you become overhydrated, this is also not a good thing. This can start to damage your liver. And you can actually start to feel lightheaded as a result of drinking too much water. Most of us don't drink enough water. We should be drinking about 3 liters of water a day, which is probably way more than most of us drink. So most of us don't have to worry about being overhydrated. But this is what homeostasis means: maintaining a constant and stable internal balance. All living organisms regulate body functions and substances. For example, pH, water, glucose, meaning sugar, temperature, as well as many others. Our brain is going to control this balance involuntarily. We don't actually have to think about it. You don't wake up every morning and tell your body like, hey, I feel a little feverish today. Can you cool it down? Thanks, I appreciate you. Your brain already does that. When you've got a little bit too much of something, your brain can start to trigger you in ways to get rid of some of it. When you've got a little bit too little, your brain can trigger you in ways to get more of it. For example, when your body is low on water, you feel thirsty. That's your brain telling you, hey, our system is starting to run low here. Can you get a little bit more for us? When you've got too much sugar, you get a little bit of a rush, a sugar rush. And this is your body saying, hey, we've got too many carbs in our body. In order to avoid storing those carbs as fat, we got to get rid of some of them. So you need to get up and start moving around a little bit. So your brain is controlling all of these functions without you even having to think about it. When you start to sweat, that's your body saying, hey, it's too hot. You need to start to cool down. When you get goosebumps, hey, it's too cold in here. You need to find a way of creating some energy, creating some heat. We've talked about this before, but pH is a measure of how much hydrogen is in a substance. And that tells you how acidic that substance is. How strong of an acid is that substance? Really acidic substances have lower pHs. For example, 1 or 2 or 3. Your stomach acid is really about a 2. In some cases, it might even be more acidic. It might be a 1. Pure water has a pH of 7. And then there are things that we would consider basic or alkaline, which have higher pHs. Ammonia, bleach, soap, these have higher pHs. So our cells are needed to constantly regulate our bodies. We know that this is because of enzymes too. Does anybody remember what happens to an enzyme when it is placed in an unideal condition or in an unideal environment? What happens to that enzyme? What happens to enzymes when, wonderful, Jason, thank you. They will denature. They will literally, their shape will change, which means that they will no longer function. This is an important word for you guys to remember. So excellent job on that one, Jason. If the shape denatures, then the enzyme is not going to work. And this can happen when, let's say you've got an enzyme and it likes to be at a pH of 4, but you put it in something that has a pH of 9. That's not going to work well for the enzyme. Our body has many different enzymes and they all prefer specific environments. Some like more acidic environments, some like more basic environments. So your body has to be able to maintain balances, even in different places of your body, in order for those enzymes to still function properly. Within our blood, the range that can be considered healthy is really, really small. The blood needs to be really between 7.35 and 7.45 in order for things to be okay. If we go even up to 7.8 or 7.9, it's going to be so unhealthy that your body can die. You can die. If it goes so low as 6.5, again, you can die. So our blood has to maintain normal level of pH. I just show that to show you how sensitive our bodies are, but also how really amazing and remarkable our brains are that they can do this process of homeostasis without you even having to think about it. Okay, so homeostasis is a process that occurs at a cellular level. And homeostasis is facilitated by the cell membrane. The cell membrane controls what enters and exits the cell. So this is how your cells can strike and maintain a balance, excuse me. The cell will let certain things in and also let other things out in order to maintain a balance. Homeostasis occurs at the cellular level. Now, the cell membrane facilitates this process of homeostasis, and it has some really special properties that we need to focus on. So I would definitely write down each of these highlighted properties. The cell membrane is a phospholipid bilayer. That means that it is made up of 2 layers of lipids. We'll see exactly what that looks like over the next few slides. The cell membrane is also flexible and fluid. That means that it can shift, it can change shapes, it can flow, and it can move. As we will see, it's important that it doesn't move too much, though. The cell membrane is considered a mosaic because it has a lot of substances that are embedded inside of it. Inside of a cell membrane, you might find 100 different proteins. You might find a couple hundred different carbohydrates that are embedded in it. These proteins might be kind of tunnels through the membrane to allow certain things in and out more easily. Or they might be receptors for cell communication. Those carbohydrates kind of are like antenna to help the cell identify its environment. Okay, and so the fact that there are things that are embedded in the cell membrane help to make sure it is not too fluid. We don't want the cell membrane moving around too much. So having things embedded in it help to maintain some of its rigidity. And then the cell membrane is what we refer to as selectively permeable. Only certain things can be let in, and only certain things will be let out. It is selective. It's choosy. It's picky. So please make sure you write down these 4 super important qualities of the cell membrane. It's a phospholipid bilayer. It's made up of 2 layers. Those 2 layers face each other. I guess they face away from one another, actually. Well, you'll see what I'm talking about. It's a flexible and fluid membrane. It's a mosaic because it has things embedded in it, and it is selectively permeable. Okay. So this phospholipid bilayer is made up of a hydrophobic region and a hydrophilic region. Hydrophobic, hydro means water, and phobic means fear of. So the hydrophobic region has a fear of water. This means that it is a non-polar region that does not like water. It does not want to come in contact with water. Picture it as oil. Oil does not mix with water. This is the same for this hydrophobic region. It will not mix with water. But the cell membrane also has a hydrophilic region. It actually has 2 hydrophilic regions. This suffix philic means love for, or filio means love for. So hydrophilic means a love for water. So these are polar substances that do like water. They enjoy being near water. They will choose to be near water if possible. Okay, so let's see what this looks like. Here we've got the cell membrane. This is what the cell membrane actually looks like. We've got a phospholipid bilayer. We've got one layer that faces kind of like this with the 2 fatty acids that look like noodles in this case. And then another layer that's facing the other way. Again, the 2 fatty acids. We've got the hydrophilic head represented by a red ball here, and the hydrophobic tail represented by these kind of yellow, like I said, they look like noodles. So this is the phospholipid bilayer. Hydrophobic tails, again, they're hydrophobic. They have a fear of water. They don't like to be near water. So they face one another. And by doing so, they kind of insulate one another. They keep the water out. Believe it or not, what we're talking about here obviously is on an extremely microscopic level. It's extremely, extremely small. So water molecules cannot actually fit through these hydrophilic heads. Even water molecules are too big to fit in here. So on the inside of the cell membrane, where there are hydrophobic tails, there is no water. So I want you guys to draw this in your notes. I want you to draw circles to represent the hydrophilic heads. I want you to draw squiggly lines, curved lines to represent the hydrophobic tails. I want you to label them. So I'm going to leave this up for about 2 minutes so you all can draw this. It doesn't need to be a Picasso-worthy depiction, but really what we need to focus on are the labels, or is the labels. 2 minutes. Okay, that was 2 minutes. So of course, if you have not finished drawing, you will be able to go onto YouTube and see this recording, the recorded lesson a little bit later. So please don't forget about that YouTube channel. I want to see some of the views going up. I know some of my honor students have been watching the videos and they have said that it's helpful just to be able to go over your notes again. But keep that in mind that is available to you. And I will find the link and send it to you right now. So we've got about 9 minutes left in class. We are going to finish up soon. All right. So you can see here how the cell membrane moves a little bit. You can see that the balls that are at the top, the blue balls here, they are the hydrophilic heads. They do like water, so they face water. On the outside of the cell, it's an aqueous environment. And also on the inside of the cell, aqueous just means that it has water. It's made of water. So these hydrophilic heads face water. The hydrophobic tails, which look like strings here, they face the inside. They don't like being near water. So there is no water on the inside portion of the cell membrane. Okay. Nothing to write down here. We're going to talk more about this, but there are protein channels that are found within the cell membrane that help to transport larger materials through the membrane. Some of them have specific jobs. Some of these protein channels only help water. For example, what we can see here, these are water molecules that are moving through the cell membrane. Again, water cannot go through the cell membrane by itself because the inside of the cell membrane does not like water. So it's just not going to work. Water needs the help of essentially a tunnel in order to get through the cell membrane. And these tunnels are made of proteins. All right, so I do want you all to write this down. Carbohydrate chains help to stabilize and identify the cell. and protein channels help move larger molecules through the cell membrane that cannot move on their own. All right. So there is this concept of concentration, which is a measure of how much of something there is in a given volume. Concentration is a measure of how much of something there is in a given volume. Some possible units: grams per milliliter. You don't need to write these units down, just giving you an idea. Grams per cubic centimeter. In chemistry, you'll learn about something called the mole. So a mole per liter is a specific type of concentration called molarity, or pounds per gallon. But just more generally, concentration is typically mass divided by volume. And again, it's a measure of how much of something there is in a given volume. So let's look at these two containers. They have the same volume, meaning that they are the same size. They take up the same amount of space. But they have different things inside of them, as we can see. So I want you guys to tell me which of these has the greater concentration of yellow circles. The container on the left or the container on the right? Which one of them has the greater concentration of yellow circles? Thank you, Jason. We can see that the container on the right has 7 of these yellow circles versus only 3 on the left. So the container on the right has the greater concentration. What about those purple squares or diamonds? Which one has the greater concentration of those? Good, the left. Again, we've got 6 on the left and only 4 on the right. So they're close in concentration, but the cell on the left has a greater concentration. What about of these smiley faces? Which one has the greater concentration of the smiley faces? Yeah, in this case, they have equal amounts. And there's a phrase that we use for that equal amount that we'll see on the next slide. But oftentimes, the cell membrane is able to establish a concentration gradient. The cell membrane, of course, controls what goes in and what can go out of the cell. So in controlling that, the cell membrane can keep equal amounts on both sides. Or it can say, I want to have more of this substance on the inside than I do on the outside. And it can take in more. Or it can say it's beneficial to the cell to have more on the outside than on the inside. So it can start to, allow some of those substances out. But just in general, A concentration gradient refers to a difference in concentration between one solution and another, such that one is more concentrated than the other. So in this case, the more concentrated solution is outside of the cell. It's in the extracellular space. The less concentrated solution is on the inside of the cell in the intracellular space. Eventually, the cell membrane is allowing movement of whatever this substance is, this blue hexagon. It's allowing them to move across the membrane, and it will establish equilibrium. Eventually, they get to the point where there are equal amounts on both sides of the membrane. Equilibrium is when the concentration of molecules is equal on both sides. When the concentration of molecules is equal on both sides. Give me 1 second. All right, we're not going to finish today. That's okay. We have a couple slides that we'll need to go over again tomorrow. But because it's 11:34, I just want to remind you guys that we do have an exit ticket per usual. There are 2 assignments for today. One of them is U4D2, the carbon cycle, and the other one is called U4D2, interactive cell membrane questions. So I know you've got this 30-minute break now. You can use it as a break, as you know, you deserve that, your lunch break. Or you can use it to knock out at least one of these things, the exit ticket and an asynchronous assignment. Of course, all of our asynchronous assignments will be due on Friday, February 19th, so this Friday. So keep that in mind, folks. You do have time to work on these assignments. We did have one on Friday that you should have done, but if not, it's too late. So don't let these assignments pile up on you. Do a little bit every day, and it will go a long way. With that, thank you all for being here. Thanks for participating. And well, the few of you who did participate, I appreciate you. Everybody else, I will talk to you. I know some of you do have appointments today, so I look forward to talking with you later today. Kadaria, you and I are supposed to be talking right now, so you can remain on this call if you want to. And everybody else, I'll talk to you later. Actually, Kadaria, I'm going to stop recording. We will go to another call.